Makasih ya.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, we are from group 2 and we want to presentation about control disease of orange plants. First, let me introduce our member. My name is Nur Hofifah with study number 59, Fujiyanti with study number 62, Annisa Erikawati with study number 168, Luhung Dharma with study number 188, Desty Dwi Artanti with study number 233, Ilham Faiza with study number 239. Next, the background. Citrus plants are annual plants that have been developed in Indonesia around 70 to 80%. National leading fruit commodities whose existence spread almost throughout the territory of Indonesia. In poorly maintained gardens, the rate of attack already indicates a heavy category. This can result in the death of natural enemies and encourage environmental damage and blast pest population in the future. The next, citrus include an important group of plants belonging to the Rutaceae family. Their commercial importance is enormous, and their fruits, very rich in vitamin C, are not only consumed fresh but also transformed into juice, wedges, preserved jellies, and jams, as well as providing byproducts for cosmetic and medicinal uses. According to Directorate General Horticultura, citrus fruit production in Indonesia reaches 2 million tons each year, while its consumption in 2018 total almost 2.76 until 2.45 million tons.
Okay, orange plant disease. Disease that attacks citrus plants of various types and ways of control are also different. First, scab. Citrus scab is a common disease experienced by rootstock seedling in nurseries, especially when citrus as rootstock are all orange. Infection occurs by an inoculum consisting of conidia and possibly ascospores from the scab and the fruit. Symptoms can occur on leaves, twigs, tillers, and fruit. The new scab in a mixture of fungus and host tissue, which is a raised surface that is pink to light brown in color. Scab disease control. The first, regular pruning, a seedling in dry land using glycoliadum. Chemical control is carried out by spraying rootstock nurseries in mature, susceptible plant using protective fungicide with active ingredient of copper, and by using systemic fungicide with active ingredient of binomial or carbendazim, especially before plants sprout.
Okay, next is product disease or diplodia. Diplodia or product disease is the main disease that causes death in the stem and branches of citrus plant. The spread of this disease in Indonesia is almost in all citrus plantations, especially those that are more than 10 years old, with less intensive maintenance. This disease is caused by a fungal infection, Diplodia theobromei. Next. There are two types of diplodia. The first one is wet diplodia. The symptoms of wet diplodia are indicated by the presence of golden yellow gums on the affected branch or twigs. In advanced stage, the plant skin peels or can even lead to death. And the second one is dry diplodia. The symptoms of dry diplodia are more difficult to recognize that at the beginning of the attack because there is no product which is a hypersensitive reaction of infected plants to localize pathogens so that they do not develop. In advance or several attacks, the break or brains will peel off. They then dry out or even cause death. Next, diplodia disease control. We can do maintain the cleanliness of the garden by burning plants, especially dried wings and brains that have been attacked by disease and then immediately burned. And then maintain the cleanliness of agricultural equipment by burning shears, knives, and other agricultural tools must be washed immediately after use and then simmered with a cotton that has been moist with 70% alcohol. And the last is coating. Coating trunks and branches with California plum.
And then CVBD disease. CVBD or citrus vine flame degeneration is one of the most important diseases in citrus, which has grown widely and has become a major obstacle to efforts to increase citrus production. The cause of CVBD disease, which is also called citrus greening or young long bean, is Liberobacter bacteria. CVBD vector. The spread of CVBD disease from one plant to another is carried out by the vector insect D3Q or Homoptera psyllidae. The spread of CVBD disease in nature depends on the population density of DC3 as an insect vector and the presence of an inoculum source. Transmission process. Pathogens can be transmitted by insect vectors from one plant to another after passing through acquisition feeding period, and then inoculation feeding period, retention period. In acquisition feeding period, vector to feed on diseased plants to get the pathogen. Inoculation feeding period, vector to feed on healthy plants until it can transmit the pathogens. Retention period, which is the time interval between vector can still transmit pathogen.
Next, GVPD symptoms. In external symptoms, first, in young plants, a presence of buds that develop slowly. Second, plants usually produce poor quality fruit. And in mature plants, the most common symptom is branch with yellow leaf, and the last, leaf on the infect branch protrude like a brush. And in internal symptom, the leaf-borne pollen of diseased plants is thicker than the leaf-borne pollen of healthy plants. In the leaf-borne pollen of diseased plants, there are thick wood cells which are pathway from near the sclerenchyma to near the xylem. The cheek wall is several layers of pressed cell wall, and in various tissues in the leaf there is an excessive accumulations of fine starch particles. Next. Uh, And for picture E, partial attack rate, in these conditions, most of the branch or twig
of the plants are still healthy, so that control effort by burning the symptomatic plants part and branch with good maintenance can still save the plants. And for picture B, C, D, E, the plant has been completely attacked. In these conditions, the concentrations of L. asiaticum bacteria in the plants was already high. And for picture E, advantage symptom of GVPD attack in these conditions, the plants must be eradicated. Next.
Next is CVPD control. How to control the CVPD disease? First of all is raising CVPD citrus seedlings. The planting of CVPD citrus seedlings is an absolute requirement for successful citrus planting effort. And then next monitoring of CVPD symptomatic plants. The purpose of monitoring is to analyze the ecosystem and find out CVPD effects as an early warning and determining early stage control activities. Next is eradication of affected plant. Eradication of citrus affected by CVPD is an effort to eliminate diseased plants, both citrus as the main crop and other Rustaceae family plants such as Kemoni. Next is our last step is vector insect control. Until now, DC3 is the only insect that transmits CVPD. So the presence of DC3 in citrus plantation should receive serious attention. Next. Next is conclusion. Orange or citrus SP is one of the national leading fruit commodities whose existence spread almost throughout the territory of Indonesia. In poorly maintained garden, the rate of attack already indicates a heavy category. The key to successful citrus farming is not only dependent on superior seedlings, but also strongly influenced by site selection and preparation and crop maintenance. Diseases that often attack citrus plants are scab, diplodia, and CVPD. The disease cause a lot of damage and loss in citrus cultivation. Control efforts that can be done are implementing integrated disease control such as using resistant plants, garden sanitation, eradication, using biological agents, and vector insect control, and many more. Okay, thank you for your attention. If there are any question, you can drop in the comment section on Spada or in WhatsApp group. Assalamualaikum warahmatullahi wabarakatuh.